and I have to talk on new SGLT2 inhibitors, clinical evidence and therapeutic positioning. And in the US and Europe, they have ertigluflozin, which I'm not going to dwell with. And in India, we just launched a new SGLT2 inhibitor, which is actually a very old SGLT2 inhibitor called remogliflozin. So what I'm going to do very quickly is that we know diabetes has exploded in the world and India is right up there in terms of its progression. And we also know that diabetes has very strongly got linked with kidney and heart. And we have moved away from that insulin resistance paradigm of insulin sensitizers, obesity, and quickly we have moved into this whole SGLT2 bracket. And all these current therapies have their own challenges. Gliptins, with all its challenges, still have this pancreatitis is still there. It's like, though I put a question mark there, angioedema urticaria is still very much there. Insulin and sulfonylurea still have hypoglycemia, weight gain. Carolina is still not published, though whatever you heard from the previous speaker clearly shows that there is a statistically significant weight gain and hypoglycemia and lack of durability from the Carolina of the sulfonylurea molecule. So I don't think it's still standard of care. So Dr. Umperez was very right. He still will keep his fingers crossed because they still cause hypoglycemia, which is statistically significant. They still cause weight gain and they are not durable. Metformin has its own challenges. Few patients will be always intolerant to metformin. Glitazones can unmask weight gain, edema, heart failure. And even today in the US, the FDA label on gliptins that it increases hospitalization for heart failure has not been removed. And even in the Carolina trial, there was a hazard ratio of 1.21, which means linagliptin actually increased heart failure by 21%. People won't talk of that. Now, kidney is the focus. We know that renal gluconeogenesis is important. We know that compared to non-diabetic individuals, there is a kidney contribution to glucose. When somebody gets diabetes, kidney's output for glucose goes up by 300-fold. So there is renal gluconeogenesis, a very clear, well-known physiological fact. Secondly, compared to normoglycemic individuals, when somebody becomes type 2 diabetics, the SGLT2 activity goes up very clearly. And the third is the renal threshold. Clearly, there is an increased renal threshold in the renal tubule. So that's something very well known. So in normal renal threshold is 180 and it shifts to the right when one gets diabetes and it can go right up there. But what SGLT2 inhibition does is actually moves that renal threshold from the right to the left, and that's something which is very well known now. And fundamentally what people are doing is that they are restoring the kidney physiology by SGLT2 inhibition. So one thing which is very clear, and this is very clearly illustrated by many nephrologists across the world, including Brenner and his group and several other nephrologists who very clearly feel that there is this hyperfiltration hypothesis in early stages of diabetic kidney disease. And clearly it's linked to the increased renal sodium glucose resorption and decreased sodium delivery on the macula densa. And very clearly we know that very well now. And the minute we give SGLT2 inhibition, this hyperfiltration is reduced. And there is probably a tubuloglomerular feedback which occurs. And this tubuloglomerular feedback, which reduces the hyperfiltration, fundamentally improves the sodium at the macula densa. So all you do is a switch in the kidney which had less sodium to the macula densa, which was there in early diabetic nephropathy in the hyperfiltrate state, now has adequate sodium or increased sodium delivery at the macula densa, and you get natriuresis and glucouresis. Remember, all SGLT2 inhibitors are smart diuretics. Independent of the company you choose, they all work. And they all have positive outcomes on heart failure. So one clear-cut thing is they work like smart diuretics and they work. We have a very well-known autosomal genetic condition of SGLT2 gene, which is called as familial renal glycosuria. And clearly, there is a good glucose which persists in the urine. In face of a normal fasting glucose, normal GTT, no renal tubular dysfunction, normal life expectancy, no complaints, no hypoglycemia, no hypovolemia. So remember, genetically we do have a condition which exists, but one thing is clear-cut beyond reasonable doubt, that the cardiorenal protection SGLT2 inhibitors give is clear cut. There is absolutely no doubt whether it is through the glucose-urea pathway or natriuretic pathway. Clearly, beyond reasonable doubt, it creates a state of negative calorie balance, uric urea, blood pressure drop, volume drop, and clearly cardiorenal protection has been established beyond reasonable doubt. If you look at any organ, cardio protection is universal. And I think if you look at any SGLT2 inhibitor across board, they all confer cardiac protection. Only thing is the way we study them is different. And probably there is very strong evidence which is emerging that they also protect the kidneys, which I just alluded to. And clearly the intraglomerular pressure drops, the blood pressure drops, progression to CKD drops. So clearly if you see from that standpoint, the SGLT2 inhibitors for primary and secondary prevention of cardiovascular and renal outcomes in type 2 diabetes. This was a meta-analysis of all the CVOTs published in the Lancet, clearly shows us that this is a class effect which is very well documented. In fact, you can see here the drop in cardiovascular events, CV death, heart failure, progression to renal disease. Clearly you can see that this is seen not only just in CVOT trials, but also in the real world trials. If you see all the real world trials, again the data and the verdict is very clear cut. But when we actually study how these agents were discovered, they were from apple bark trees. We know that apple bark tree has a natural bicyclic bioflavonoid, which was in 1933 isolated as fluorescein. Fluorescein was a non-selective SGLT1 and 2 blocker, and it could not be developed as a proper drug because it had a significant gut metabolism, so it got metabolized very fast. But a prototype of fluorescein was developed by chemical researchers and chemists, medicinal chemists, and they looked at O and C glucoside molecules which improved stability and SGLT2 selectivity. So the O glucoside, fundamentally two molecules got developed, remogliflozin and sergiflozin, and both these molecules could have seen the light of the day if GSK had continued that research. While the C glucoside was the canagliflozin, dapagliflozin, empagliflozin and ertugliflozin, while the O glucosides are short-acting molecules, the C glucoside are the long-acting molecules. And therefore, we had the first canagliflozin, probably one of the strongest SGLT2 inhibitors on planet Earth, works very well, has excellent data on CV and renal outcomes. Then came DAPA, developed first, but launched a little later. In Europe, actually, it came in 2012, but across the world, it came in 2014. The DAPA, again, an excellent agent, very good data again on heart failure and declared TIMI and renal outcomes will come soon. And then came EMPA. EMPA came with a very big impact because they just got a bonus trial from a CV outcome study called EMPA-REG, and they were able to piggyback that trial very well because it was launched in 2014, and right in 2015, they had a trial which they could back up. And then, of course, ertugroflozin got launched by Merck. GSK actually started its SGLT2 program somewhere in 2003. And 2008, they diverted all their funds in vaccines because after 2007 disaster with the Evendia. And they had developed a compound which has been worked in the Europe and US called remogliflozin. But the people who developed it later on formed a small group called Kisai. Kisai outlicensed it to Glenmark, which has commercialized it in India. We all know that SGLT2 inhibitors are endorsed by all treatment guidelines, whether it's ADA, ESD, and we know its consensus algorithm very well, and we know that they are clearly there in every guideline. In fact, ACC has gone one step further on them, ACC-AHA has gone one step further on them, and they have arrived. Let me now quickly take you through the journey of remogliflozin, which is available as now a new SGLT2 inhibitor. What is available in India is remigliflozin etabonate. It's an ester prodrug of remogliflozin. It is a selective inhibitor of renal SGLT2 365/1. It's a O-glucoside. It is a meal-independent SGLT2. It is used as an indication for type 2 diabetics in adults. It was originally discovered in 2002 by Kisai. GSK outlicensed it from Kisai. In 2009, GSK completed its phase 2 trials and discontinued it due to strategic reasons. 2011, it got commercialized for operations through another pharmaceutical agreement, and Glenmark got interested in the product in 2016. when they went to the DCGI and they told them to do phase 1, phase 2, phase 3 trials all over again. And they did all of them, and they completed their phase 3 trial, and now they are also doing an ongoing CV outcome study for the same. So prior to that Indian study, there were 26 clinical studies involving 25 countries, including 2,000 subjects. And one of the lead investigators in this was Bob Henry and Sundar Mudaliar from UC San Diego. And there is data on this compound from different ethnicities: Japanese, Hispanic, Caucasians, and Indians. So there were 22 phase 1 studies done on the compound, whether they were the human compounds, the drug interaction studies, the formulation studies, the specialized studies on cardiac repolarization, renal insufficiency, type 1. In fact, type 1 data is published in Diabetes Care by Dr. Mudaliar's group. And then there were 3 phase 2 studies on 800 patients and 1 900 patient phase 1 study, which had been a head-to-head comparison with DAPA for safety and efficacy, which is a pivotal trial based on which the DCGI in India has approved the drug. So what is the pharmacokinetic profile of remogliflozin? Because now it's available in India, we'll be using it. And from the Indian data, and this is under publication now, clearly remogliflozin itabronate gets deesterified by the esterases in the mucosal wall, and the active moiety is remogliflozin, the GSK 189074. It gets converted into the GSK 279282, which is active metabolite, and fundamentally, if you see, the exposure of the drug has been given after single dose. It's a prodrug. It's completely and rapidly absorbed in the duodenum and jejunum. It's 100% bioavailable and no effect of food on the plasma exposure, so it's meal independent. It is immediately converted to remogliflozin by the esterases in the mucosa. It appears in plasma in 15 minutes. The Tmax is around 30 to 45 minutes. It's 65% bound to the albumin, no accumulation with repeat dosing, and no differentiation or preferential distribution in the blood cells. It is extensive metabolized through different pathways, including CYP and non-CYP pathways, and none of them have any meaningfully well-known drug-to-drug interactions. In fact, their drug-to-drug interaction studies have been quite extensive, both done by GSK in the Western world and in India by the Glenmark Pharmaceuticals. It's excreted with a plasma half-life of the active drug being around 2 hours and of the metabolite being around 4 hours. So biological half-life being 3 to 4 hours. And it's something which is very well known. Again, you can see the steady state plasma concentration with BID profile is consistent, and the free levels are throughout inhibiting the SGLT2 inhibitor with a KI throughout the day and night. So with the BID regimen, a good level and inhibition of renal resorption is possible and plausible, and this is the CGM data which is available for the 24-hour profile in type 2 diabetic subjects. I am not presenting the type 1 data here at all. The Indian trial which was done for its registration for phase 2 was a twice-a-day study which was randomized, and that was a dosing study with a head-to-head comparison with 30 mg of pioglitazone, and it was a 12-week study. And it was using 50 mg twice a day, 100 mg twice a day, 250 mg twice a day, 500 mg twice a day, 1000 mg twice a day. This is the data which is seen very clearly, very robust data, and that allowed the DCGI to allow it to pass through the phase 3 efficacy data. The phase 3 was a randomized, double-blind, double-dummy, parallel group, multi-center, actively controlled study for safety and efficacy in type 2 diabetic subjects. There were 58 sites, and it was a head-to-head study with DAPA, 100 mg twice a day and 250 mg twice a day. And now they commercialized with 100 mg twice a day. It was a 24-week study with A1C being the primary criteria, type 2 diabetic adults 18 to 65 years, stable on metformin with A1C between 7 to 10% with safety endpoints. And it was a head-to-head study with DAPA, and you can clearly see the data is extremely consistent and comparable in terms of A1C reduction. In fact, if you see here, the A1C to baseline, DAPA 0.4, 0.49, the 100 mg BID dose, and 0.63, the 250 mg BID dose. And when it goes further for 24 weeks, you can again see that both the 100 and 250 mg doses of remogliflozin are statistically non-inferior to DAPA, that was the comparator, on the A1C reduction. And clearly you can see here, the database is very clear-cut on the A1C reduction standpoint. It works very efficiently, very well when it compared to the DAPA arm. and achieving the A1C2 target also. Both the Remo groups, whether it was the 46% or 48% with 100 milligram BID or 250 milligram BID, it was pretty consistent. And again, the need for rescue medication, again, lesser subjects needed less rescue medication with Remo compared to the DAPA arm. And again, fasting blood glucose was consistent. The change of progression over 24 weeks was consistent. Prandial reduction of glucose was again comparable and consistent. And the multi-point profiles were also consistent. Body weight reduction again was very consistent. And the blood pressure drop was consistent. So clearly you can see beyond reasonable doubt, this drug in terms of efficacy and safety works. Let us look at the adverse events of DAPA and remagliflozin in the registration trial. They were very, very similar. There was no major deviation of any of these compounds. The lipid impact, the female genital mycotic infections, the male genital mycotic infections, the urinary tract infections. Again, the adverse events were very comparable with DAPA, which was used as the gold standard drug, as a comparator drug in that. The eGFR drop also is very consistent. So summarizing, basically now we have a new SGLT2 molecule available in the Indian market. It has been intensively researched molecule in 26 clinical studies globally, and also has gone through a phase 1, phase 2 and phase 3 program in India, and has been launched now in India, and has now actually commenced a Remo CV study, which will be available to us probably 3.5 years from now. But as I said, it stands to logic, and they have commercialized the drug with 100 milligram twice a day, not the 250 milligram twice a day, because they did not find any major clinically meaningful benefit between 250 milligram twice a day compared to 100 milligram twice a day when it was compared head to head with DAPA. And the fasting PP, A1C reductions, weight, blood pressure reductions were very consistent. And the non-glycemic arms, which we look at, weight, blood pressure, were very comparable. Adverse event profile was very comparable, and clearly it offered a cost benefit. So from a cost economic standpoint, it offered a benefit, and efficacy, safety being similar, and of course heart failure data on reduction being consistent. We can see that we have now one more agent which we can use and which we can try out on the Indian patients. Thank you for a patient hearing.